the truth is, the whole reason why I'm even coming out with this is I realized that there are certain therapies and antidotes to snake venom that perfectly take care of stopping the death and carnage from what they call SARS-CoV-2 viral infection and all of its variants. And I need to make sure those doctors worldwide know this is what you have to continue to use. It doesn't matter what the variant is that they call it. It's always the same solution, always. Because
once you realize you're treating snake venom,
everyone around the world needs to know this. You need to picture every COVID-19 infected patient as if they've been bitten by a snake.
Got it.
And then address the envenomation. If you really want to get real technical, take every COVID-19 patient that tests positive for COVID-19 with a PCR test or anything else, or a blood test, and run an ELISA IG test, which is what they use to test for snake bite in ER centers. But just run it, it'll be positive.
You know, snakes inject using fangs, using a puncture, a subcutaneous injection of snake venom. And that's exactly what the vaccine is.
Exactly.
It's a simulation of the snake.
The hypodermic needle was made from the design of a fang.
Right. Oh my gosh. No wonder they're trying to kill you.
Oh yeah. Okay, here we go. Ready? Let's go on. Come on. Let's go. Next slide.
Oh, okay. Really? There's more?
Oh yeah, there's more. In February of 2015, just like remdesivir, check this out. The organism of envenomation group also showed, these are mice given crude cobra venom. The organism of envenomated group also showed alveolar hemorrhage and myonecrosis after 6 hours of envenomation with the cobra venom.
Wow, this is exactly what they're seeing in patients.
Exactly what they're seeing. All right, here's an actual picture of the slides of the lungs of rats.
Same one?
Yep, same one.
Okay.
And you'll see here, A, the image from the control pulmonary tissue, this is normal, showing it's all normal. B, lung tissue of envenomated group indicated extensive tissue damages and showing inflammation, cellular infiltration, and alveolar hemorrhage. Next slide. Edematous swelling of lung tissue with subsequent accumulation of pulmonary infiltrate blocks the alveolar air spaces, disrupting gas exchange and lung mechanics, leading eventually to what?
Yeah, respiratory failure, sure.
What do you see with SARS-CoV-2? All you hear about is respiratory failure.
Especially after remdesivir.
Especially after remdesivir. What if it's venom? And they're pumping this venom inside of you. It's insane. In conclusion, I wrote, is this ironic or damning? It can be concluded that the exposure of cobra venom encourages serious histopathological alterations, disease changes on renal,
kidney,
and pulmonary tissue, and moderately on intestinal tissue of envenomed mice. Therefore, further studies need to be carried out. Blah, blah. Next. Did you know that cobra toxin, an actual thing, is a cardiotoxin, heart toxin? So it says down here highlighted, cardiotoxin is a peptide toxin produced by the Chinese cobra. It is cytotoxic also. What does cytotoxic mean?
Kills cells.
Kills cells. Next slide. Further into this document from February of 2022, cardiotoxin binds to the cell membrane and depolarizes cardiomyocytes, heart cells. It also shows lytic activities on many other cells, including red blood cells. It also targets the mitochondrial membrane and induces mitochondrial swelling and fragmentation, binds to the integrin alpha V beta 3 with a moderate affinity, and inhibits protein kinases.
Wow.
Okay, this is significant. A couple things. Cardiotoxic, and inside this highlighted area, it actually targets mitochondrial membranes. Next slide.
That means it's going into the cells, not just poisoning them from the outside.
Heart cells, ready? Check this out. This is October of 2021. Potential cardiotoxic effects of remdesivir on cardiovascular system. Let's go there to the highlighted part. Remdesivir can also induce significant cytotoxic effects in cardiomyocytes that is considerably worse than chloroquine's cardiotoxic effects. Remdesivir-induced cardiotoxicity is due to its binding to human mitochondrial RNA polymerase.
It's exactly the same as the snake venom.
It's exactly the same as king cobra venom that I just read from you that was published in February, 3 months after this.
Unreal.
Yes.
It's literally the exact same effects.
Yes.
On heart tissue. Okay.
Next slide. I just want to reference the Indian cobra stuff. And I want to talk about what's inside, again, the cobra study published by Gene and Tech. that Gilead bought some facilities from. I'm going to read this together because when you put together the correlation of, is it possible that SARS-CoV-2 is really just snake venom? Watch this.
Okay, is this the right slide?
Next slide.
one. Okay.
Currently, snake antivenom is the only treatment effective in the prevention or reversal of the effects of envenomation. Since 1896, antivenom has been developed by immunization of large mammals, such as the horse, with snake venom to generate a cocktail of antibodies that are used for therapy. Okay, so they say since 1896, you take snake venom, inject it into a horse to be able to make antivenom. This is what they describe in Nature magazine. Next slide. I couldn't believe I even found this, Mike. You're going to freak out. This is in Brazil, Costa Rica. I found this on Getty Images, not a joke, where you buy stock photos.
Yeah, right.
They have a description on here. I want to read it together.
This is unreal.
This is unreal. Ready? Let's start with let's just read the whole thing. A scientist manipulates a velvet snake to extract venom at the Clodomiro Picado Institute in San Jose, Costa Rica, September of 2020. The Clodomiro Picado Institute obtained coronavirus proteins from laboratories in China and the United Kingdom to immunize 6 donated horses for treatment, in which plasma was used to produce a viral treatment to be tested in 26 patients infected with the new COVID-19.
Oh my God, that's the same. That's how they make antivenom for snakes. I mean, against snake venom.
It's the only way.
It's the only way. So when they say they obtain coronavirus proteins, it's venom, which is a protein.
Snake venom protein.
Snake venom protein. And they did the same. And the picture's a snake.
Pictures of a snake. All right.
It's almost like they accidentally told the truth for this image.
So start with this part here. Read that last
To
sentence for me.
develop the drug, the Institute, based on its half-century experience in the production of antivenom, which it currently exports to other Central American countries, Ecuador, Colombia, Peru, and several African nations.
This institute got SARS-CoV-2 proteins injected into a horse. These people have been for half a century specializing in one thing.
Making antivenom.
Making antivenom. Is that not insane?
Okay.
I thought that was insane. January 2021, snake venom phospholipase A2 possess a strong virucidal activity against SARS-CoV-2 in vitro. Wait, what? Snake venom phospholipases A2 possess a strong antiviral activity against SARS-CoV-2 in vitro. I just find that interesting. Then in January, same month, 2021, venom from one of Brazil's largest snakes could reduce COVID's ability to multiply. That's interesting. I thought it was a virus. Next slide. All right, now I want to ask you something, Mike. Next slide. Mike, I want to ask you something. Did you know that during COVID-19 in 2020 and 2021, did you know that worldwide there were 2 global crises going on at the exact same time?
No.
British Medical Journal.
Bite crisis? Are you kidding me?
British Medical Journal, Global Health. Look, read it. Snake bites and COVID-19. Read the title. 2 crises, 1 research and development opportunity.
Because it's the same stuff.
Because it's the same thing. Read the highlighted part down
crazy.
here. Read this down here.
Despite inherent differences, snake bite envenoming and COVID-19 have much in common in terms of research and development challenges and opportunities. Unbelievable. I had no idea that this was being linked, even in the mainstream media. This is British Medical Journal.
Right. Virus, snake venom. 2 crises, but only 1 research and development opportunity?
Right, because it's the same. It's just rebranding.
It's the same thing.
Oh my gosh.
I couldn't believe it. How about this? March 2021. In Business Wire online magazine, a Berkshire Hathaway company. 1.58 billion antivenom market, global growth trends, COVID-19 impact and forecasts. March 3rd. I had no idea. Look at this. There is a significant increase in the number of deaths reported due to snake bites during COVID-19 pandemic. Really? I thought we were all locked down.
Exactly, yeah.
Did you know
How can there be more snake bites?
Did you know? It even says in here that Texas, I think it said, oh yeah, let's read it. Keep going. This is creating a substantial demand for antivenoms. More than 350 snake bites were reported in Texas in 2020, which is an increase of 40% over the value registered in 2019, as per the June 2020 article titled Texas Snake Bites Increasing During COVID-19 Pandemic. I thought we were all locked down in 2020. Did you know anybody got bit by a snake?
No.
I don't know anybody. And I'm in Texas. We're both in Texas.
Yeah, I mean, I saw a lot of snakes, but none of them bit me. You don't play with them is the key. You just don't play with them.
Just don't
you
play
don't
with
mess with them.
Isn't this odd that there's this Berkshire Hathaway's reporting on the fact that there's a booming business now in anti-venom. At the same time, because of COVID-19, we're getting snake bites during a period of lockdown. It's insane. It's all because it's related. Remember, next slide,
Like
Arizona said what?
The body.
It's just like it because it is. How about this? February 2021. Snake venom derived bradykinin potentiating peptides, a promising therapy for COVID-19.
But wait a minute. How can these snake venom derived molecules treat COVID if COVID is based on snake venom?
Right. Remember, I told you there are vipers that have different components that act on different systems. So the blood clotting effect of one venom going into your body, you could use the other one that's anticoagulant.
I see.
Got it? This is why the crate and the cobra venom is tied together as SARS-CoV-2, because cobras have a increasing prothrombin time. Do you know what that means? Increasing how long it takes for your blood to clot. That is a blood thinning, hemorrhaging effect. Crates do the opposite. They create blood clotting and thickening.
Oh, wow.
Just saying.
So wait a minute. This would also explain why the more vaccine injections people get, the more their immune function is destroyed.
Absolutely. There is tons of evidence supporting that snake venom destroys T cells, CD8, CD4 cells, and that's what we're seeing reported around the world with one shot after another.
Right. And now they're pushing the fourth shot, even in America and in Israel and the UK. And they say the antibodies, the so-called antibodies, are gone after 4 weeks instead of, a year or forever as they used to claim. Okay.
Very true. So in February 2021, The Guardian reports that there's new advances in COVID-19 therapies. You want to take a guess at what it is? Look at this. Highlighted. First paragraph.
Snake venom enzyme. Oh my gosh.
As a therapy for COVID-19. Really, I just want to give you a glimpse that this is what people are talking about in the media, but we're not paying attention to. Next slide.
But metformin, also a blood thinner that causes hemorrhaging if you overdose on it, right?
Exactly right.
All right.
All right. Snake venom can stop COVID-19 from multiplying. Oh, no way. Researchers in Brazil have discovered that a particle in a venom of a pit viper venom has the ability to stop the reproduction of coronavirus. Since when? Never. Next slide. But the same article. Quote, we were able to show this component of snake venom was able to inhibit a very important protein from the virus. Oh, let's look at
the coagulation protein.
Exactly right. All right, so let's look at Wall Street Call. Look at this. Next one.
Okay, next one.
Wall Street Call, December 2021, just a few months ago. There's this anti-venom market future growth outlook. Look who's on the list.
Are you kidding me? Merck and Pfizer?
And Pfizer.
They're in the anti-venom market?
Pfizer's hugely in the anti-venom market, and Merck has been in it for years. Do you know that lisinopril, the high blood pressure drug made by Merck, is made from snake venom?
You're kidding me.
Nope. They have a patented snake venom that's an ACE inhibitor that helps to lower blood pressure, all derived from snake venom.
Unreal.
Next slide. Now, it says Pfizer on that previous slide has huge potential for growth in the anti-venom market, or venom market. This is November of 2021, just a month earlier. Pfizer files for an emergency use application for COVID-19 drug Paxlovid.
I see that.
All right, I want to read the last paragraph in this write-up. Next slide.
Okay, unreal.
Go ahead and read the whole paragraph there, Mike.
Really?
Yep,
okay.
Go ahead.
This drug inhibits cysteine protease from the PA clan proteases that play coagulant role as they're involved in blood clotting cascade. As a cysteine protease inhibitor, it may disrupt coagulation and have blood thinning qualities leading to predisposition of internal bleedings. Several snake venoms also belong to PA. And what is PA again?
So PA is a clan protease inhibitor. And they define it throughout there.
What does PA stand for?
I don't even remember.
Okay, but several snake venoms belong to PA clan proteases and interfere with blood clotting cascade. So you're right. This is the anti-clotting venom. Now, this is interesting in light of presence of neurotoxin-like motifs noted earlier in the SARS-CoV-2 sequence related to snake venom neurotoxin superantigens. Oh my God, it's all right there.
It's all right in front of you. All right, next slide. Let's keep going. Now we're going to talk about some of the solutions. Venom-derived neurotoxins target nicotinic acetylcholine receptors.
Oh, I've seen this come up in the literature.
This was huge in the beginning of SARS-CoV-2. Do you remember that around the world, hospitals were reporting that amidst this acute respiratory distress syndrome they were calling SARS-CoV-2 or Covid-19, hospitals around the world were reporting this odd thing they were noticing. The least demographic of all people hospitalized with SARS-CoV-2 or Covid-19 were smokers. They said it was less than 5% around the world were being hospitalized for Covid-19 who were smokers. It was the least.
That, yes. It was the nicotine.
And as they were actually reporting this is not a joke. They started reporting this, and up and through May, they were reporting on it. Our FDA, NIH, and CDC immediately went into action in May of 2020 and started telling everybody in the media, everyone in the world needs to know the highest, most likely demographic group to end up being hospitalized for COVID-19 were smokers. They flat out lied to everybody and said, now there's never been a better time than to quit smoking.
Oh my.
Because
in this case, the nicotine could have saved you.
Absolutely. These nicotine acetylcholine receptors, if you use nicotine, it blocks these neurotoxins inside of snake venom from vipers. You could totally handle SARS-CoV-2 with nicotine. I'm not joking. As I've learned this stuff, people have called me, hey, doc, I'm struggling with the Omicron variant or Delta variant if they got diagnosed with COVID-19 from a PCR test that I think is bogus anyway. If they were struggling, all I did was send them to go get Nicorette gum or nicotine patches. Right. And I would tell them, look, you've been struggling for 2 weeks. Just put this stuff in there, 48 hours or less, all over with. This is the one and only thing that doctors around the world are missing because they don't realize it's venom. It's neurotoxins from viper venom. And they attack the nicotine receptors in your brainstem that control your diaphragm's ability to breathe. Wow. Then you drop your oxygen. That is what these neurotoxins do. It is not a lung infection. This is a venom poisoning. And it's getting into your brain stem through your blood supply, crossing the blood-brain barrier, which is what this venom's designed to do, paralyzes your diaphragm's ability to breathe, your oxygen levels are dropping. These people need nicotine to bind to those receptors because venom will be secondary to bind to them. This is huge. I'm telling you, everyone around the world needs to know this. Any variant that comes up in the future, you need to use nicotine.
I'm going to have to add some items to my stockpile of emergency medical supplies at this point.
That's right. I
do
not recommend
have nicotine gum yet.
We do not recommend smoking.
Right. I'm not recommending smoking.
Nope. I do not recommend it at all.
Nicotine patches and things like that.
Right. Absolutely. But that's what this venom does.
You know what? Tobacco is actually easy to grow, too. I used to grow tobacco in Ecuador.
Did you?
But it's regulated in the United States. But anyway, you can grow tobacco. You can make a nicotine tincture. Theoretically.
Is there a regulation on how many tobacco plants you can grow? Is there like a limit?
There are some kind of crazy regulations.
Okay.
But whatever.
We can explore that. Maybe something new from BrideyOnStore.com in the future.
Yeah, if we survive all the snake venom, we can figure out how to grow nicotine. Okay.
So I want to go into this study because this reads just like SARS-CoV-2. Read it. Nicotinic acetylcholine receptors are channel-coupled membrane receptors activated endogenously by acetylcholine, together with GABA, 5-HT3, and zinc-activated ion channels. You have to understand.
Right.
Nicotine and zinc is targeted. Zinc receptors are targeted by snake venom. I told Zev, I was like, Zev, he didn't even know this when I filmed this. He doesn't know what I'm working on, Dr. Zev Zelenko. But I told him, please keep harping on hydroxychloroquine and zinc. This is why they keep punishing doctors for recommending zinc. Zinc will block the negative impacts of snake venoms.
Right, and this explains the effectiveness of the zinc ionophores with quercetin.
Exactly right.
Right.
Yep.
Oh my goodness. And right, so the media was attacking zinc, attacking everything that worked.
Yep.
Okay.
So let's just stop here for a second before I even go any further. I just want to touch on a few things that everyone needs to be focused on. Every person treating COVID from here on out. It doesn't matter what the variants, more are coming. They're not finished with their agenda. Nicotine is number 1. You need to consider nicotine supplementation. I don't care if it's in a patch, a gum, little packets you can dissolve underneath your tongue, like people dip tobacco. There's a little nicotine packets you can just put in there. It's not actually tobacco. Nicotine,
really?
these patients need to be taking. 2, zinc they need to be on. Number 3, they need to be on hydroxychloroquine or ivermectin. These are zinc ionophores. I actually prefer hydroxychloroquine.
Really?
Yep, absolutely. If I'm right, on the remdesivir emergency use authorization, they only talk about one drug that cannot be co-administered with remdesivir. They don't say ivermectin. They say hydroxychloroquine. They say it's an antagonist to the antiviral properties of remdesivir. Hydroxychloroquine and anti-malarial drugs, which are antiparasitic drugs, are proven to block and inhibit snake venom poisoning. And it's been that way for like 100 years. And I'm not joking, like 100 years. It's phenomenal. So you have nicotine, zinc, hydroxychloroquine, or ivermectin if you want to. I prefer hydroxychloroquine. NAC to block the blood clotting factors. Some people say, what about aspirin? Well, they're using several forms of venom. You don't know if you're getting the anticoagulant, and if you're taking aspirin, it's going to make it too thin.
Right.
And then you could have internal bleeding and hemorrhaging as a result. So I'd be careful with the aspirin part. So NAC, anyway, that's what you're going to start with. That's plenty. That'll actually take care of all of them.
But the crate snake is a blood thinner.
No, blood clotter.
Oh, that's the clotter.
King Cobra
and
is
the cobra is the thinner.
Oh, you know what's amazing? Yeah. Remdesivir's emergency use authorization says remdesivir has a side effect of increasing prothrombin time, which means it's thinning your blood and you can't clot as fast.
Okay.
King Cobra has the exact same side effect.
Right.
And it says you have to check prothrombin time before you start administering and after. No one does that, but they should.
Wow.
Okay. All right, so let's get into this next part. This is kind of fun. All right, so now I want to get into touching on some of the aspects and concerns I have about the mRNA vaccines, the current COVID-19 vaccines, and particularly the mRNA ones, and their possible tie to the fact that the mRNA is most likely snake venom mRNA and not viral genetic snippets. Ready? So mRNA COVID-19 shots, unbeknownst to the majority of the world that you and I live in, who do we credit? Who's in the media all the time credited with creating the mRNA vaccines?
Dr. Malone.
Dr. Malone. He's not the one that invented this stuff. The mRNA vaccines are actually credited to 2 co-inventors. And their names are Drew Weissman, and Catalin Carrico. Let's just Carrico.
Yeah.
And they both work at the University of Pennsylvania.
Okay.
All right, so these 2 are credited with creating the mRNA novel COVID-19 vaccines. Got it? Work at the University of Pennsylvania. How long have they been doing research with mRNA vaccines? Here's one back in 2009. All right, so incorporation of pseudouridine into mRNA yields superior non-immunogenic vector with increased translational capacity and biological stability. I don't even want to talk about it or describe it. I just want you to pay attention to mRNA and then the first and last name listed here as authors on this research study. It's the 2 people at the University of Pennsylvania who have been credited with creating COVID-19 mRNA vaccines. Okay. Inside this study, they actually state in 2009, a likely contributing factor to the enhanced translation observed with modification is an increase in biological stability of the mRNAs. Indeed, higher resistance to hydrolysis by phosphodiesterases from snake venom and spleen has been reported when uridine was replaced with blank in dinucleotide substrates. So they have been using snake venom properties to create mRNA vaccines as far back as 2009. Next slide.
To achieve the stability that they need for the mRNA to maintain its
all about stability
while it's penetrating the cells.
Exactly right. We'll touch on that too. So here we go. Acknowledgements for this study in 2009.
NIH. And NIAID, that's Fauci and Collins.
You ain't lying. Next slide.
Oh my gosh.
Gets better. Next slide. I want you to read this out loud. These are the 2 authors.
Oh yeah, KK, what is it, Carrico?
Catalin Carrico
and
Drew Weissman
have formed a small biotech company that receives funding from the NIH.
To explore what?
The use of nucleoside-modified mRNA for gene therapy.
For gene therapy.
Gene therapy.
Do you know they've constantly said that this isn't gene therapy?
Right, but it is gene therapy.
The two people credited with these mRNA vaccines have been working solely on gene therapy using mRNA technology. They even have a whole company to explore it, funded by the NIH.
Okay.
Since 2009. Next slide.
Yep.
Nucleoside modifications. This is in 2011. Same two people are listed here as authors. You'll see the last two names here. Drew Wiseman, Catalina Carrico. At the University of Pennsylvania, highlighted. Working with RNA. Let's go into the study. Next slide. Highlighted here, you'll see they used G-coded dynabeads. Just remember this. Keep going. Now, in 2012, unrelated to these two people that I was just talking about, I need to read this to you because it's significant when it comes to the mRNA vaccines for COVID-19. The unusual stability of mRNA in snake venom reveals gene expression dynamics of venom replenishment. This is in 2012. Next slide. Inside the abstract, it reads, here we exploit the unusual stability of mRNA in venom to conduct for the first time quantitative what? PCR to characterize the dynamics of gene expression of newly synthesized venom proteins, not natural venom peptides, synthesized. So they're creating these. Next slide. Inside the same article, but on a different platform, the stability of mRNA in venom is biologically fascinating, still 2012, and could significantly empower venom research by expanding opportunities to produce transcriptomes from historical venom stocks and rare or endangered venomous species for new therapeutic, diagnostic, and evolutionary studies. Next slide. Inside this study, they actually take snake venom, different components, SVMPs that are actually in SARS-CoV-2 research studies, PLA2s that we talked about are phospholipases, which are actually the actual dangerous components of SARS-CoV-2 that elicit morbidity and mortality found in cobra venom. Remember that? All right, and then they find this DNA light up here, okay, the stability of this stuff. They actually do it with PCR amplification with snake venom mRNA. All right, next slide. I have to show you something. Next slide.
Okay.
This is snake venom DNA from 1984 in 2012. When they titled this Unusual Stability of mRNA in Snake Venom, they're finding that snake venom stored since 1984 till 2012. Look at this. They found these inhibitory peptides from mRNA were isolated from a venom sample extracted and lyophilized in 1984. That means there's 30 years worth of stability of this mRNA, and they're shocked in this study that the venom properties of the snake don't destroy the mRNA over time that it's stored in. They can't believe the mRNA survives all of it because they're degenerative and destructive.
But we're told that the mRNA is stabilized by being capped with things like polyethylene glycol and things like that. Is that just a cover story, or
Yep.
From what this is showing, it looks like the snake venom is the preservative for the instructions, and if the instructions themselves are also how to make snake venom, right?
Yep. So in 2012, they just published a study that they found this unusual stability of mRNA. It even lasts 30 years by itself, not degraded by snake venom, other components. And then in 2014, 2 years later, I don't know if the slide's in here, in 2014, they actually found they could even make it more stable, mRNA from snake venom, by wrapping it in a nanoparticle hydrogel.
Oh,
not a joke.
okay.
And then they combined it with dynabeads in 2015.
Is that a brand name of some kind of nanoparticle? What
Yeah,
is that?
I'll show you in just a second because it explains your mRNA shots. I'll show you in just a second. All right, so here we go. mRNA extractions from venom. Polyadenylated messenger RNA was purified from lyophilized venom using dynabeads, mRNA direct kit. Dinyl intravigen using the manufacturer's protocol. Next slide.
Well, so we can just buy a Dynabeads mRNA kit, and we can.
Yes, you can right online. There's a company called Thermo Fisher Scientific that makes it.
Oh, Thermo. Yeah. Okay. Say no more. All right.
Say no more. Yep. All right. So here we go. Real-time PCR. I just want to reference here. This is how they actually magnify the gene sequences of snake venom is using PCR. What are we using to find SARS-CoV-2 around the world that's never been used for a virus before? PCR testing. Next slide. These results demonstrate that potentially large quantities of mRNA of high quality can be reproducibly recovered from snake venom. More than adequate amounts of mRNA were recovered from each venom sample for downstream PCR analysis. This is in that same study. Next, our demonstration that mRNA can be detected in venom at each time point during the complete time course of venom synthesis is remarkable, because we would expect snake venom glands to present a highly unfavorable environment for mRNA preservation due to the diverse array of destructive nucleases and phosphodiesterase and naturally acidic conditions. In an extreme extension of this investigation, we also report, and this is important, we also report that mRNA encoding snake venom metalloproteinase. Do you know what metalloproteinases do?
No, I'm not familiar with that.
They actually make your cells deplete metals like zinc, copper out of your body, which you will then start to suffer from strokes, hemorrhage, inability to breathe. All right, metalloproteinases, serine proteases, C-type lectins, Kunitz inhibitor, all of which you can do any research study that SARS-CoV-2 does all of those. You want to know why? Because it's snake venom. All right. An inhibitory peptide was PCR amplified from this venom, which was extracted in live flies in 1984. They are shocked in 2012. They could still see mRNA amplified with PCR in the venom is still actually bio.
A lot of these vaccines are lyophilized powders that have to be diluted.
No kidding.
With solution.
Just like every venom
so
and
you're saying they're
antivenom.
just shipping out powdered snake venom, calling it a vaccine, lining people up to inject them.
Guess how remdesivir comes? In a little glass jar of lyophilized powder, just like snake venom does.
And that stuff's going to last for 30 years.
Easily. It's not even stored in snake venom for 30 years. It's just freeze-dried.
Okay.
All right, next slide. This is August 2011 mRNA study continuing. RNA containing was cleaved efficiently by RNase. RNase T1, RNase T2, nuclease P1, and snake venom phosphodiesterase. Although there is some indication that pancreatic diesterase and snake venom phosphodiesterase may cleave RNA with reduced efficiency. Let's look at snake venom phosphodiesterase. Ready? Next slide. You can actually buy snake venom phosphodiesterase right online. It's $133. Snake venom phosphodiesterase. Purified lyophilized. If you would like to do your own mRNA research, just buy some of this stuff. And you can cleave your own mRNA. Next slide. On the same page, I need you to understand this. This is very significant. When you order this stuff and you're a research lab wanting to cleave RNA with snake venom phosphodiesterase, it actually states that this phosphodiesterase from snake venom is inhibited by reducing agents such as glutathione,
which is also being removed by the FDA.
Yep. NAC produces glutathione. Selenium I talk about helps the liver to make glutathione, and they do not want you buying NAC.
Right. At
all. But this is why you're seeing such great success with glutathione injections. Like people that go around and do IV injections of glutathione, they see miraculous results with COVID because of this. But look at, you see cysteine, and then what else?
Ascorbic acid, vitamin C.
Ascorbic acid.
Yeah.
Which I already showed you the study where cobra venom was literally chopped in half, the morbidity and mortality with vitamin C concentrations in plasma. There was a direct correlation to survivability from SARS-CoV-2 and vitamin C loads and venom peptides called phospholipases.
What's shocking, completely inhibited by 5 millimolar EDTA. That's a very low concentration.
Yes. This is why they're trying to block this stuff.
I mean, 5 millimolar is, I don't know, a few drops in your body or something?
Yes.
I mean, it depends on your blood volume, I guess, but...
Isn't that interesting?
I'd heard of people taking EDTA. I'm not recommending, I'm not saying to take it. I'm just saying I'd heard of people taking it. Suddenly, it would deactivate
snake venom phosphodiesterase.
Exactly.
Which is what they're using to make mRNA. Next slide. Guess what funding was for that research study I was just quoting? You see National Institutes of Health, and then at the bottom you'll see, is there a conflict of interest with doing this study? Well, Katalin Karakov and Drew Weissman have formed a small biotech company. This is in 2011, separate study. The National Institutes of Health funds them to explore the use of nucleoside-modified mRNA for gene therapy. Okay, 2011. All right, now, I'm about to now show you just how in your face this is, okay? Pediatrician Dr. Peter Hotez, January 2022, creates a new vaccine. Next slide. And now in February of 2022, could a patent-free vaccine offer a COVID solution that stands up against Alpha, Delta, Omicron, and all future variants? This is the one Dr. Peter Hotez in Houston. From obscurity to a Nobel Prize nomination, Houston scientists acclaim for their patent-free COVID-19 vaccine. Next slide. These two are the two. Microbiologist Maria Bottazzi, her colleague Peter Hotez, and their team at the Texas Children's Hospital Center for Vaccine Development last month unveiled Corbavax, the world's COVID-19 vaccine. And doctors say it could be a game changer. Next slide. This is what it looks like. I want you to tell me how that reads. I mean, look at the label. This is a COVID-19 vaccine. I want you to
Cobra.
I want you to transpose the B and the R.
Cobra. Vax.
It's actually a play on Cobra vaccine.
Right.
Now, interestingly, Hotez and this other lady are up for being nominated for a Nobel Prize for this new vaccine they created. The only problem is they didn't create it. Biological E created it. But I'm really curious why that is they got the credit. Next slide. January, same article. Well, just to clarify, the producer of the vaccine and the owner of the vaccine is Biological E in India. Corbavax is their vaccine. Okay. Why are they being nominated for a Nobel Prize when they didn't even make it? Next slide. Now, this is in the India news. I went to go find it. I wanted to see, has Biological E been passing this out already in India? COVID-19 vaccine Biological E's Corbavax gets EUA for use in the 12 to 18-year-old age group. This is February 22nd, 2022. About the same time, these two in Houston are getting credited with a new vaccine being nominated for a Nobel Prize called Corbavax, which is really Cobravax. Next slide. In the very first paragraph, it says, Biological E, a Hyderabad-based vaccine manufacturer on Monday, received EUA from India's drug regulator for their COVID-19 vaccine for now the 12 to 18-year age group. The Drug Controller General of India had already approved this vaccine, called Corbavax, for restricted use in emergency situation among adults on December 28, 2019.
That's before COVID was even being talked about.
This was being used before COVID was even talked about in December of 2019. All right, so I just want everyone to know, these people in Houston did not create this vaccine. Biological E did. They created a vaccine and called it Corbavax, which when I saw the name, I was like, oh my God, it reads Cobravax. And I went just like this. I'm going to go to Biological E's website and I want to see what they manufacture. Next slide. I'm sure it's just a coincidence. What do they produce? Next slide.
You're kidding me.
This is their product catalog.
Snake antivenom.
They make snake antivenom. First one is cobra venom. Next slide. Common crate venom.
Crate venom.
Next slide. Biological E's homepage or website. Vaccines and biologics. Corbavax is India's first indigenously developed protein subunit COVID-19 vaccine. I would love someone to test this from the manufacturer of cobra venom. I'd love for you to test that for cobra peptides or proteins. And I bet they're injecting into every India child now 12 to 18 years old, along with adults, a
Just injecting
snake venom
them with snake venom.
vaccine. Did you know about this?
No.
Next slide. Read the title.
Moderna co-founder using mRNA technology to treat venomous snake bites.
This is July of 2021. It's almost absurd once you put this lens on that everything's related to snake venom. Everything related to COVID becomes very clear how to treat it, how to handle it, and just how bogus it is.
So you're saying the vaccines are snake venom, basically. You're saying the mRNA is instructions to tell your body to make more snake venom, and you're even saying that SARS-CoV-2 itself is derived from snake venom.
All right, so this is a great point. I'm going to stop right here. You ready? Because we're looking at Moderna's co-founder, whose only drug they've ever made is the mRNA COVID-19 vaccine.
Right.
In July of 2021, he says we can use the mRNA technology to treat venomous snake bites. Okay, Mike Adams, I want to ask you a question. Have you heard of Dr. Hoff in British Columbia, Canada?
Yes, Hoffe, Hoffe.
Yep. Have you seen where he said he had all these patients who all of a sudden experienced, after getting their mRNA vaccines, they started experiencing like long haulers COVID fatigue.
Yes.
They couldn't walk 100 yards. And he decided to do a test on all of them. Do you remember what that test was called? I didn't even know what it was when I heard it.
Oh, yes. Was that the microclot test?
Do you remember what it's called?
No, I forgot the name of it.
It's called a D-dimer test.
Yeah, D-dimer.
Okay. Now, when I say I wanted to see is there a relationship to everything related to COVID, what's being reported to COVID, and snake venom, is it possible, I thought, let me just look it up and see. Is there a connection to D-dimers, the D-dimer test, and what are medical doctors trained to look for if they see elevated D-dimers? So I'm going to read this out loud to you. How are elevated D-dimer levels interpreted? And this is March 2022, this year, last month, on medscape.com. Very reputable online medical journal. I want you to read, there's only 5 bullet points on the page of what medical professionals and lab technicians are trained to recognize when you see elevated D-dimer levels. I want you to read the 5th one.
Snake venom poisoning. These are the possible explanations from elevated D-dimers.
If you see elevated D-dimers, one of the things you're trained to look for is snake venom poisoning. And I am absolutely certain that there are snake venom peptides being used as these mRNA injections, and they are reacting with coagulation throughout the body. Venoms from snakes have this massive amount of creating micro blood clotting all throughout the body. And Ryan Cole has talked about this nonstop underneath his slides as a pathologist. He sees all this micro blood clotting throughout the body.
Right.
It is a reaction, I'm certain, related to cobra venom.
And the organ damage,
snake
the damage
venom.
of organ tissues on the surface of the lungs and the heart tissue, liver, pancreas, kidneys.
I couldn't believe this
ovaries.
was. I couldn't believe this was online.
I'm telling you, it's.
Couldn't believe it.
See, but what you've done here is amazing, because once you know what terms to search for, and by the way, I mean, I should tell the audience, everybody go out and start searching on these terms now, because believe me, the internet's going to get scrubbed of anything that says snake venom, vaccines, anti-venom, all of this.
There's a
the
reason why
scrubbing will happen immediately.
There's a reason why I have thousands of documents, and they're all downloaded, saved, and put on hard drives. And they're printed. I mean, you could take away all my hard drives that have it saved in all multiple places around the country, and then you have to burn all my documents that I have printed because I printed them all.
Well, they may try that.
They might.
You
Who
should
knows?
Distribute the documents as quickly as possible as well.
But I have to tell you, I was very nervous when I came and talked to you about this a couple months ago. I was very scared.
Yeah, but you didn't have all this research at that time.
I didn't have all of this. I just told you, I think I'm onto something and I don't know what to do about it. Anyway, it was great. Anyway, I just want you to know, the greatest part of all of this is everyone around the world's going to know what to treat these people for. It's been very complex, and they don't know what to look for. Everybody who's had an mRNA vaccine who has any long-term side effects needs to go do this D-dimer test. Go to LabCorp, go to Quest. You can order it yourself. Go in there, pay for it, and get it. And if you have elevated D-dimers, one of the things you're looking for is snake venom poisoning.
So wait a second. So for those who have the mRNA injections and they've had the transfection alteration of the DNA of the cells, their cells are producing then, essentially what you're saying is they're producing a snake venom molecule that's circulating in their blood. Now, it's also going to show up on the surface of their skin. They're going to shed snake venom. We're talking about humans being turned into serpents.
Have you heard of people shedding their skin after getting these shots? I have. I saw it on Stu Peters.
Yes.
Jeff Jackson. You want to know why you're shedding your skin?
Because
like a snake.
Like a snake. But I mean, this idea. I mean, the demonic elements here. If Satan were to try the ultimate prank on God's creation, he would try to turn you from human into a serpent. And that's what this is doing.
It's exactly what this is.
And then you run around shedding venom onto other people. And I can't tell you how many people I know who they get headaches around vaccinated people, or they get nauseated around vaccinated people. And there are some women who have infertility and strange menstruation effects when they have been around vaccine shedding people. Suddenly, this all makes sense. You can't have a baby, you can't have a regular period if your body is infested with venom.
Can you imagine the gravity of this information right now and the impact it's going to have on the pharmaceutical industry as the world finds out?
I mean,
could you imagine they're about to approve injecting children with these mRNA shots,
infants, yeah.
like 6-month-old to 4-year-olds.
That's right.
And they're already doing it to 5 to 11-year-olds. Thank God only 25% of American children have gotten them in that age group. But could you imagine as a parent now thinking they could be injecting your children with snake venom? Who would want their children to ever get bit by a viper? None. You're going to sign up and take your children to volunteer to get it injected into you? What about your elderly? What about those with comorbidities? The peptides inside of snake venoms actually target comorbidities or inflamed organs that are already present. They destroy those that are diabetics. They're targeting diabetics, heart failure patients, kidney failure patients. I mean, how many people in this country are on dialysis right now?
Oh, yeah, hundreds of thousands, I think. It's a
insane.
lot.
Insane.